Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Giacomo Cavalli from the Institute of Human Genetics in Montpellier on this show. Giacomo, please let me briefly introduce you to our audience. Your academic journey began with a biology degree from the University of Parma. You then earned your PhD from the ETH in Zurich between 1991 and 1994, focusing on nucleosome positioning in yeast under the supervision of Fritz Thoma. Subsequently, you pursued postdoctoral research at the University of Heidelberg from 1995 to 1998. And in December 1998, you then established your laboratory at the IGH in Montpellier, where you have been conducting research ever since. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Oh, this is a good one. In fact, I happened to become a biologist because I was supposed to become an economist. I did secondary studies in economics, and I realized that I really didn't like it. And there was a time when I was a bit lost. I didn't know what to do, and it was just a friend. We discussed a lot with friends and mates and so on, and she told me, yeah, but why don't you try and look up this stuff in genetics that is happening? And it was, at the time, it was the 80s. It was the beginning of cloning, and making plasmids. And so I was starting to read Scientific American, and I really got fascinated in that. And then did a little bit more reading. And then I gave it a try, which was not supposed to work because usually when you work in economics, you're skipping a lot of mathematics like integrals and stuff that you use for, science, hard science and biology. But I caught up and finally it worked out.
So does your interest in economics still help you in running your lab?
Oh, yeah, that was a thing. Yeah, it did come back. I took some responsibilities, also some management responsibilities at some point for the institute, and it did help. I'm not so sure. I'm happy that it helps because management is maybe not my favorite thing. So in fact, I confirm that I love science, rather than managing. But yeah, it's certainly helpful.
Yeah. So let's come to your science that centers, and I guess I caught it up correctly, around Polycomb and 3D genome organization and TADS. So let's start with your work on Polycomb. Can you take us back to the time when you started your own lab? How did you feel and what did you want to do and achieve in your own lab?
Yeah, it was actually the whole trajectory I had in a way is sort of self-consistent. So when I was at university already, there was one of the big questions, like how much information there is in the sequence of DNA and how much would perhaps be outside or or maybe connected to components that regulate DNA. And of course, at the time, there was not many tools to enable to study that. And initially, I was thinking more of information that might be contained in architecture. That is, maybe the DNA itself as a sequence, is important. But then in addition, there are some structures of DNA or maybe of chromatin that themselves contain information that really one could not address for a very long time. But then, of course, you could think that if you find the components that can impose alternative states and maintain these states, maybe let's say through cell division, even in the presence of the same DNA sequence, that would already be a great start. And then what could these components be? And so, at the time of my postdoc, we started to have, there could be candidates that could be heterochromatin proteins, where you have more or less of them present on heterochromatin and then have more or less silencing. And Polycomb components were another set at the time of proteins that were supposedly different from heterochromatin, but still able to shut down chromatin locally and in a cell type specific manner or in a time specific manner. So I thought, okay, we don't know what's the architecture, but at least we know the proteins when we can see if we play with them, if we can have alternative states. And so that was kind of at the end of my postdoc. So then the dream I had was, all right, can I go back now and understand whether these proteins also play a role in the architecture of chromosomes? And so somehow we connect all the dots. So that was the idea to try and do that in the lab.
So what you did in the early phases of your lab, you was working on maintaining or on the inheritance of chromatin states. So how would a state be inherited to daughter cells, for example? So in this context, the Fab7 is well defined in the cellular memory module. Yeah.
Mm-hmm.
So how does this work or how is this regulated in the context of Polycomb?
Yes. So in fact, so the idea was, so we know that there are elements, and this is what the one you mentioned, this FAB7 is one of them. They are target elements for Polycomb components. But again, we know that their target genes in some cells are off, in other cells are on, and we didn't know why, what decides this. So it was hard at the time to know exactly what transcription factor could be involved and how to manipulate them. And also, if you manipulate them, then you're going to have, big phenotypes, maybe lethality. So what we tried to set up was a sort of synthetic system where we would have a transcription factor that is a yeast protein, so it doesn't affect the life of flies. We were doing all the work in flies. And this would have binding sites in a transgene. The transgene would also contain our FAB7 element, and we knew we could identify binding of Polycomb to this element in the transgene. So without the presence of the transcription factor, Polycomb proteins go there, bind, shut down the element, and also a reporter gene, which was placed just downstream. But if we now would express a pulse of transcription factor, what happens? And we showed that a pulse of factor can transiently eject Polycomb components, so it can reverse the silencing. And then the question we could ask was, okay, so if we do that as a pulse, and then we wait longer, what happens? Is the element reacquiring Polycomb and shutting down transcription again? Or if we move the system towards a very strongly active state, maybe the state is maintained even in the absence of the initial transcription factor. It was the experiment. So, and we showed that we could actually indeed switch the element from a silencing mode into a maintenance of activation mode. And later on, we also identified what could be proteins that help that, and that was the tritarch components. Some people call them compass components now because they have been also identified in mammals. And so what happens is that the factor is binding. It is inducing transcription. When it does, it removes Polycomb. And now we have these tritarch proteins that help maintaining the transcriptional states. In fact, what we also found was that Polycomb indeed is able to bind back. So it's binding, but now it can no longer shut down. So that was nice. But then what we realized also quickly after that, and that was done in the lab, was that this factor was establishing contacts in the nucleus with other binding sites of Polycomb. And in particular, if you would have multiple copies of the same sequence, these would be highly preferred. So somehow these elements set up a sort of barcode, maybe of binding proteins that really like to find each other. If they don't, then they will also go to some other Polycomb binding sites. And so they set up an architecture in the nucleus. And I could show that this architecture is in particular helping to maintain the silent state. So when you have silencing, this involves establishing a lot of these contacts. The active state probably doesn't need it so much. I mean, you more like need transcription factor that start transcription. So, and then that is how we actually went back into the nuclear organization, initially with imaging, and later on with methods like IC and this kind of like mapping contacts genome-wide.
Yeah, covered quite a large time frame of your work. How does DSP-1 play into the recruitment of Polycomb group proteins?
That is a piece of a puzzle which, in fact, we thought we completed, but we now realizing we have not. So that is a protein that is one of the multiple components that recruit Polycomb. In flies, there are proteins that bind DNA, which we and others showed that help recruiting Polycomb components. The fact is that Polycomb complexes do not have sequence-specific binding to DNA, but they are recruited to specific places. So, then the question is who recruits them? There is not a single factor that is able to recruit them alone, but there is a cooperation between multiple DNA binding proteins. So one is this DSP-1. There is another one that is called pleiomeotic that has actually a famous mammalian counterpart called YY1. There is another one called GAGA factor. There is, and then a couple more. There's an SP1 protein involved as well. So there are 4 or 5 factors. What we did with this DSP1, at the time, people already had identified 4 and GAGA factor. And, they thought, okay, if we have multiple binding sites of them, they will be enough to recruit Polycomb. And we did a few tests and we did not find that. So then we basically started adding up some other binding sites and making a synthetic element. When we added the binding site for DSP-1 on top of those that were already known, we could show that now Polycomb binds. That's very nice, but now we went back actually. It's not published, but we went back to some of these binding sites, but now in vivo. And instead of making transgenes, we try to go to a given locus and then we synthetically deplete or delete or add binding sites individually. And it's proving to be very tough because even the combinations of factors that we thought are sufficient now don't necessarily work. And that is, so in fact, the grammar is not completely clear. But yeah, I think the end is still a combination of factors, but I think we are missing maybe 1 or 2 so when you go in chromatin, which is more repressive, you probably need a series of binding sites before you can really recruit robustly Polycomb proteins.
What about RNA?
And RNA, yeah, that's also another very controversial thing because we didn't work much on RNA. There's a big controversy in the field where some people contend that RNA is helping recruiting Polycomb, but other people show data that suggests the contrary, that in fact RNA is stripping away Polycomb. So the thing is that I said there's no sequence-specific DNA binding activity, but some of the Polycomb subunits do have nucleic acids binding activity. And this is more like a charge. So whenever you have a charge, negatively charged nucleic acid, it will interact, and in particular RNA does it very well. So it could be that when you transcribe, in fact, this RNA would strip away the Polycomb complexes that now would have the RNA targeting in addition to the DNA target that they were bound to. How to show who is right, who is wrong? Probably, I mean, what it looks like is there are interactions, but they are not very strong. And so it's maybe the balance of interaction that you have in a given locus and the amount of RNA that is present that will ultimately define whether you maintain the Polycomb complex in place or whether you take it out because now you have a lot of RNA and that is actually taking out Polycomb. So It doesn't seem that there are highly sequence-specific, RNA-dependent sequence-specific activities that recruit Polycomb to a target. Of course, I say today, but maybe tomorrow we will change our mind. But it's more like a mass action effect that depends on the kind of sequence you have and how many RNA molecules you have, as opposed to the DNA target that is there. So,
Okay. So more recently, you looked at the tumor suppressor function of PRC1 proteins and that this can take place independent of PRC2. What did you find there?
Yeah, so this is also quite intriguing. So people found this tumor-linked activities of Polycomb in mammalian systems first. So already in the 90s, there were some works, including some work from Martin van Doorhuizen in the NKI in Amsterdam. They were showing that Polycomb can cooperate with MYC to induce, actually Polycomb is BMI1, which is a subunit of a canonical PRC1 complex. And this one, when overexpressed, can lead to cancer, can cooperate with MYC to lead to cancer. There were other people extending this data. They looked at different subunits. Some subunits started to look more like tumor suppressors than oncogenes. So things looked a little bit complex. And in mammals, you have multiple paralogs for each of the subunits of these complexes. In flies, the situation is much more simple. So we thought, well, let's look in flies where we can look at any single mutant. And usually when we take out a subunit, there's no, compensating subunit that can be expressed. There's a single gene. So we will understand the situation much quicker. So we try to look systematically, and initially we started with PRC1 subunits, and when we delete them, we get tumors. It's usually ectodermal dependent tumors. So they start from tissues that are basically ectoderm. We are extending it now, but so you can have tumors out of different tissues. When we then started looking at PRC2, we got the surprise because there we didn't see any effect. And so we tried to understand why does it happen? Because we have done a lot of ChIP-seq experiments in embryonic tissues, and we saw a lot of colocalization between the proteins of PRC1 and PRC2. So then it was hard to explain. And then we started looking, well, what about if we take the tissues that we are studying, which were more advanced stages of development? And all of a sudden, we started to see that the binding diverges between the two complexes, or at least some sites, which are the canonical sites like Hox genes or PAC6 or, hedgehog or WINS, they stay always together, but then there's many, many other genes which start to be bound by PRC1, but not PRC2, or by different levels of the proteins, or by maybe both of the complexes, but PRC2 is catalytically inactive in some of the targets. So you have PRC1 bound, you have the ubiquitination activity of PRC1 that is present, and PRC2 is either not bound or bound but doesn't trimethylate lysine 27. And instead, then we found that many of these places that you have acetylation of lysine 27 and the genes are expressed. So that really got us very excited understanding what happens there. And we found two categories of genes, some that of these where PRC1 is bound and are active. In fact, binding is still repressing. It is modulating the expression. And this is where those genes where Polycomb binds at the promoter only. And these are like frequently cell cycle regulator genes. So it's proliferation genes. And so that is basically, it's tempering a little bit the proliferation of the cells. And then there is a second class of genes where now Polycomb binds to the promoter, but also to enhancers of these. And these are also active genes. But then in that case, the binding to both elements is favoring the interaction of inesors with promoters, and these are typically differentiation genes. And if you take down Polycomb there, you actually reduce the interaction between inesors and promoters, and you, in fact, lose expression of differentiation genes. And so these two activities seem to cooperate to induce tumorigenesis.
Yeah, it seems to be a very complex system that is in place there. However, and I want to switch gears a little bit in the interest of time and focus on 3D structure of chromatin. As you mentioned earlier, or as we mentioned earlier, next to Polycomb, you also focused on the three-dimensional folding and functional organization principles of the Drosophila genome. And this was exactly the title of a Cell publication from the year 2012, where you produced a high-resolution chromosomal contact map derived from a modified genome-wide chromosome conformation capture approach that you applied to Drosophila embryonic nuclei. So what did you learn from this, and how can this be connected to the other papers or the work that has been done in that space?
Yes. No, yeah, in fact, so that was, so the idea there when we started this project was still to understand Polycomb. We suspected that these proteins are forming a lot of long-range interactions, but we didn't have the full network, so we wanted to understand that. Initially, we did not think we would discover much more. We probably thought, okay, we're gonna see maybe heterochromatin interactions as well. And so we did this genome-wide. So at the time, people were developing methods. So we developed a sort of IC, but it was not exactly the same. It was similar to the Decker variant of this. And anyway, but what we found was that yes, you have preferential contacts between Polycomb sites, preferential contacts between heterochromatin sites. We saw telomers that are also getting in contact. But then all of a sudden, we stumbled into these domains that are contact domains. Some people call them TADs, some people call them chromatin domains or chromatin interaction domains or whatever. There's many names. And so what distinguishes them is that you have a lot of internal contacts. So as long as you are in the domain, everything contacts everything else pretty much. But then you have sharp boundaries and you have much fewer contacts with the neighboring domains. So you see them as sort of, if you plot the contact maps, it would be looking like triangles that are close.
I guess everybody knows it, yeah.
And yeah, so we saw this, and then we also found that you have different kinds of these domains. Some of them tend to be more repressive, so it's either Polycomb or heterochromatin types, or a third class of repressive, which is no specific histone modifications, but there's very low gene expression. So it's like sort of default silencing. And then you have active chromatin domains where you have, a good number of active genes. Maybe not all the genes included in the domain are active, but the majority of them. And what they do then is to establish long-range contacts between domains of a similar type. So active domains will tend to contact other active domains. Polycomb will contact Polycomb. Heterochromatin will contact heterochromatin. And then these sort of default guys, they actually are more spurious. They contact other domains similar to them, but they also have low-level interactions with the Polycomb or heterochromatin. So yeah, it's a sort of compartmentalization that people then also identified. They already had identified it before, when they spoke of A and B regions, basically A being active and B would be inactive. But what we found is that it's a little bit more subtle than that. There are multiple kinds of inactive kinds of contacts and active contacts. So this is something that when you look at the high-C data in memos, you also find. And this network of Polycomb contacts for us, of course, it's quite exciting because it is a little bit what we also identified by looking at individual transgenic elements and so on. And we could also show that interactions between these Polycomb regions, very long range, even across multi-megabase distances, can be regulatory, because if you start to mutate an element in one domain, you can have loss of silencing, not just of the gene that is under, close to this element, but also of genes that are located far away. And this was typically true for the Hox gene. So that was quite exciting to see.
So what you then did is you also did this similar work during mouse neural differentiation in vitro and in vivo, generating the highest resolution Hi-C maps available to this date. So can you talk about this study and what maybe about the differences between the TARDs in mouse and in Drosophila?
Yeah, absolutely. So so that basically the idea was to try and understand in detail what's happening in a mammalian system, but also like, because at the time people already started to produce pretty good maps, so we tried to improve the quality as much as we could, but also to see what happens during differentiation, because that wasn't really clear at the time. People had looked at different cell types, but not really like at a precise, homogeneous differentiation process. Try also to disentangle cell cycle differences from differentiation differences, and so we did that, and it was quite exciting because we could ask some very precise questions. For instance, what kind of specific contacts do we get? At the time, people had already shown in memos that CTCF can form loops, what people call loops, and these loops were shown to be between CTCF bound regions that are at the left and right borders of these chromatin domains or TADs. But it was not clear that enhancers and promoters can also form these sort of specific contacts. So we could find them, and we could also show that when you differentiate cells, they also change, which is much less true for the CTCF. So the CTCF was kind of, you see them, but there's not much change really in these. So then the point, but what is it good for? Maybe it has an architectural function, but that's it. And so we could find that there is also a change that is depending on functional switches. We tried to ask a question which was, are the architectural changes preceding or accompanying or following the differentiation dependent changes, functional changes in gene expression, which I don't think we answer completely. We kind of see that they accompany, so the architecture accompanies functional changes without being able to really say whether it follows or precedes, because there you would probably need a much finer time course. Certainly, we could show that there is not like a preset, architecture that is then just used by regulatory elements, but stays as it is. So there is a regulation of this architecture. And now people are getting quite excited and they are keeping on these studies. So, and we too, actually, we have improved the maps even more. It's something that we are on the process of publishing.
Yeah. Oh, I see. So we will hopefully read about that. What you then did, you moved back to Drosophila embryogenesis and identified active and repressive chromatin loops that are established with different kinetics and depend on distinct factors. So, trying to get more information about the 3D chromatin organization, basically, right?
Yeah, actually. So, and this is where I said, with the mouse study, we certainly were not exhaustive, and so we went back to flies. So we are constantly switching between flies and mammalian systems. Some of them are better, for specific questions. So, and in the fly, we recently looked at a specific loop that is regulating a gene or a couple of genes, and it's a place where polycomb proteins bind. But not only, there's a very well-known enhancer. So we could start asking questions. Are these polycomb sites contacting each other? Yes or no? What is the enhancer doing? How it is modified during differentiation or during development? And actually, that is one case where we do have a sort of architectural platform. So for some genes, and actually for many, many genes, there is a change in architecture when the gene is activated or repressed. This particular guy is a gene where you have a prominent loop between polycomb binding sites. One of them is at the promoter and one of them is upstream. And then the enhancer is more like hitchhiking on this structure. So when the transcription factors that are acting on the enhancer are absent, the gene is switched off and polycomb keeps it off nicely. But when you start to express the transcription factors, in fact, the polycomb loop is bringing the enhancer also relatively close to the promoter. And so then you have a very efficient activation of the gene by the enhancer. So in this case, the polycomb components seem to be used more as a sort of scaffold that is used by transcription factors. What we could show was that if you perturb this polycomb architecture, now the enhancer doesn't know very well what to do. It will activate its promoter, but it starts also to activate the wrong promoters. So it was counterintuitive, and I think probably not universal. It probably applies to some genes. In other genes, depending on the arrangement of elements, you might have a different output. But yeah, I think, it's always when I'm telling these things, I hear myself and I say, oh gosh, it's so complex. But, we have to regulate in a very exquisite, specific way, different cell types at different times in development. Probably this is why we have this multi-layered organization.
Yeah, that could be true. Yeah, that's another thing that you did until this time point is that you looked at like bulk cells, right? You look at the average of cells, and what is always then interesting is to look at individual cells, and this is typically done by microscopy. And you also did this, right, looking at TADs at the single cell level. What did you find there then?
Yeah, so there we try to ask, and it's something that we are still working on. The first question we wanted to ask was, well, are these TADs real? Because one could also think that, and actually people had put this hypothesis forward, that, you have preferential contacts intra-TAD compared to neighboring TADs, but in fact, it is not a very strong enrichment. If you take 2 points at a certain distance within the same domain, and then you take one of them and then a related point but outside in the next TAD, and you ask, well, how many more contacts do you have intra versus inter-TAD? It's only an enrichment of 2-3-fold, maybe. It depends on the cell types, but it's not like 100-fold. So then the question is, well, if it's such a small enrichment, is it really important? And it also, does it mean that some cells will have indeed this domain as a domain, but many other cells, in fact, don't care. Maybe they form other domains, or, you just make an average, and that's the majority structure, but it's a small majority, So, so we tried to do that, and we looked in multiple cells, and actually, in flies, it's very clear that TADs are individual domains. So TADs in flies, they are about 100 kilobases in size. And when you go in super resolution microscopy, you always find that they are really separate. So if you take probes for adjacent TADs, they are really separated, and each of the probes forms a globule. When you go in mammals, there it is also true that different, neighboring TADs are separated, but now the shape of them is much more variable between cells. And that is something where probably the nature of the chromatin that is contained in each of the TADs is important, because we found that in active TADs, they are much more, variable, and in some cells they are more open, in others more closed, that they have multiple subdomains, and in other cells they have less. And actually, if we play with the histone interactions, and that's something that you can do if you, for instance, change the acetylation status of the histones. So when you have multiple acetylation, you reduce the interactions between histones and histones long range. Now you tend to form to really break down the TADs into multiple subdomains, much more than if you don't have acetylation. So that I think is something important. And these domains that are formed, which are smaller now, it's sort of a universal thing. In every species, you can look with super resolution microscopy, you always find objects of like 100 or 200 nanometers in size that are present in the nucleus. And then in the species where the TADs are small, in fact, these are the TADs. In species where TADs are bigger, you have multiple of these nanodomains that are clustering together to form a TAD. So I think we are really dealing with what is the fundamental physical nature of chromatin in cells of eukaryotes.
Are you doing some work in this area moving forward?
Yes, because the point is, this is kind of, so I think this is reflecting really the physical structure of chromatin, but then you still ask yourself, well, is there any functional correlate of this? What happens if we modify the level of acetylation? So we change architecture, but what is the influence? And of course, acetylation can directly affect also promoters. So we would like to disentangle the effects that you can have, let's say, at promoters or enhancers from the architectural effect. And so we are trying to do that by, again, doing super resolution studies, and also we're trying to develop this multiplex fish where you can really look at the trace of chromatin in space and to try to see what counts, what is important. Do we need to have contacts between enhancers and promoters? Or also the condensation state of the polymer plays a role. If it's more condensed or less condensed, maybe with the same kind of contact, you can still change the expression status. So that's something that we are very excited in looking, and we are starting working on that.
Mm. So this already plays into my next question. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into this? I mean, now other things than what we just talked about.
That's a great question. Great question. I tend to, grants can be like, what is the biggest likelihood to get the grant? And the other approach is, what is the question that I love to ask? And, maybe it's not fashionable, but anyway, that's where I'm going. I am more, my propensity is to go with the second one. Hopefully that other people are interested as well. I think that what we need to understand two things. One is all of these, quantitative aspects. It's a tough, ask because we have a lot of regulatory components, and what we are learning more and more is that it is not a single one. So like, for instance, we see that enhancers do need to contact promoters, but you can have some subtle changes when the enhancer is switched off or switched on. So you need to have the right transcription factor binding, and then maybe architectural changes, but they are only limited. And so you need to describe it very well. But I think it's still a very important question to ask. So the nitty-gritty of the molecular mechanisms, that's one. And then the other thing is memory effects. When I was postdoc, I could show that you can have memory, but we had no clue what is really driving this memory. So what we've done recently was to show that you can have memory that can really transmit functional cellular states, not just like taking a reporter gene. For instance, we could get a tumor just based on a transient perturbation of Polycomb components that would trigger tumorigenesis. Then we bring them back, and even if we bring them back, they are not able to restore the differentiated cell state, and the cells go on to make a tumor. And we really would like to understand exactly how these tumors arise, what is the first tumoral cell, what's going wrong in this cell, what is maintained by what transcription factors downstream, etc. So, and we want to do it in flies, but also in mammalian systems.
Okay. So you're now running your lab for over 25 years, roughly. When you look back and take an intermediate summary, did you achieve the things you wanted, or would you have done things differently when you look back?
Well, I could say I achieved at least one thing I wanted. The thing that I hoped was to have an immense amount of fun and pleasure. And I think this is one of the, incredible positives of our job. And this, I tell you, it was a treat. Okay, because in science you have a lot of failures, or let's say lack of success. At least you do not. Some projects don't go as expected. You do not obtain the result that you were thinking you would, etc. But there is also a ton of, unexpected things that are really nice too, and they just trigger your curiosity even more. So it's a sort of it's just beautiful to witness this process, to live it through. So that, I think, alone is unbelievable. But then in terms of like practical goals, I think I'm happy to say that we didn't fulfill them all, because there's a lot more to understand. So we kind of, we did identify several cases of epigenetic inheritance. We could identify components. We could identify, DNA regions that are involved, architectures that are involved. But we still have plenty more to understand. So, it will probably keep busy also generations to come, not just me, because I will at some point I will stop. But
So to finish this interview, what is your personal definition of epigenetics?
Yeah, so this is a very important question. It is a definition that has changed over time. When Waddington started to define it, it was basically how a genotype gives rise to the phenotype, which is essentially developmental biology. But these things has evolved. And actually, Waddington himself, when he was asked many years later, how he felt about this controversy where people started to include the concept of memory, of inheritance, of non-DNA information, he said, "I'm fine with that. You can take it." And so I think probably the operationally the best definition would be, the molecules and the mechanisms that are able to transmit alternative states of function for a given gene or set of genes in the context of the same DNA sequence. And this could apply to transmission through cell division, but also transmission through maybe multiple generations. And it could include any kinds of components. It could be proteins, it could be RNAs. In fact, it doesn't even need to be necessarily chromosomal inheritance. When people discuss prions, for instance, or cytoplasmic maybe mechanisms where you have basically self-enforcing regulatory loops that can maintain, this could be enough. So we keep it mostly linked to chromatin components, but there's no reason to exclude other kinds of processes of inheritance.
Yeah, so thank you, Giacomo, for your time and for being on this show.
Well, thank you very much. It was a pleasure. Thanks.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.